Hey everyone, I'm very pleased to open this session by featuring you my PhD works about the biochemical flux associated to pearl farming activity and ecological consequences on the water column. I want to present that this work was supervised by Nabila Mazouni and Denis Saulnier, respectively from the UPF and IFREMER. As you may know, pearl farming activity represents in French Polynesia the second economical sector with near 8,000 hectares of exploited area. This aquaculture replicating a bivalve named Pinctada margaritifera, usually called the pearl oysters, is practiced since the 80s in French Polynesia over 25 different atolls and islands. During the last decade, important extension of the activity and punctual ecological crises in pearl farming sites have questioned the durability and ecological footprint of the activity. However, despite primary evidence, ecological footprint of pearl farming is still unknown. Among scientific questions to resolve, the interaction between pearl oysters and water column, especially its living part, the plankton, appear essential to resolve. The plankton or planktonic community is defined as the whole marine organism drifting in water currents. It constitutes the main food source for pearl oysters, but also for multiple marine species, and constitute more widely the basis of the marine food chain. In other words, plankton is a source of life in marine systems and is so essential for lagoon functioning. Interaction between plankton and pearl farming is largely associated to repeat organism activity. Pearl oysters and some bioflowing organisms, as filter feeders, catch the plankton in the water column by filtration, affecting negatively its biomass, but also the planktonic structures. After feeding process, organic matter and all organic organisms are digested and then excreted in the environment under nutrient form or pellet forms, generating so a matter flux of nutrients to the water columns. This excretion process tends to fuel the nutrient pool in the water column, which permit to sustain, in turn, the planktonic renewal. In fact, the vegetal part of the plankton, the phytoplankton, use nutrients and light to make photosynthesis and rebuild the planktonic food chain. In this way, the pearl farming and planktonic interact as a continuous turnover. It appears so essential to assess the different phases of this turnover to apprehend the durability of the whole systems. Understanding the planktonic and pearl farming interaction is the heart of my PhD projects. However, during this talk, I will feature you only the second phases of this process by featuring you first the natural flux generated by pearl oysters, secondly the consequences on water column chemistries, and finally the incidence on planktonic community structures. To study this aspect, we have selected two important farming sites which reflect in part the ecological context observed in French Polynesia, and the variability of ecological context observed in French Polynesia. At left, Arutua, an atoll of Toematu archipelago, which observed warm water all over the years and low seasonality. And at right, Mangareva, an island of Gambia archipelagos with important fragmented barrier and marked by important seasonality over the years. On both sides, multiple sampling was realized over the 2016 and 2017 years. However, during this tour, I will show you only the result from the first campaign from Arutua occurring in May 2016. To study biochemical flux associated to pearl oysters and its consequences, our methodological plan are complete thanks to two complementary approaches. The first is then an integrative approach focused to quantify directly on the farming site by scuba diving the flux generated by pearl oysters. For that, we use a metabolic enclosure, a soft big bubble, which permits to confine the pearl oyster and follow during the incubation the production of nutrients in water column. Three metabolic enclosures containing the pearl oyster are compared with an empty one used to represent the natural variability of nutrients along the incubations. Followed nutrients are split into two categories, both essential for planktonic growth. In one hand, nitrogen, including reduced form, ammonia, oxidized forms, like as nitrate and nitrates. On the other hand, we found phosphorus nutrient only shown in a single forms, easily for the end of the presentations. The consequence of this flux for water column and planktonic compartment is today with a comparative approach. For that, we compared water column in the vicinity of the farming structures, then a control site located outside the farming influence. In the water column, we'll have in the same time the nutrient concentration, like the previous related, nitrogen and phosphorus, and similarly, the planktonic community. One important originality of this work is to propose for the first time a complete analysis of the planktonic diversity. Thanks to lab collaboration and multiple methodology, including cytometry and multiple microscopy, we have rebuilt the whole planktonic structures with exceptional resolution. For an idea, more than 100 of planktonic groups are observed, but for simplify this talk, all organisms observed are clustered in 4 simplistic groups detailed here. First, the smallest one, the phytoplanktons, which include photosynthetic species, the phytoplanktons, and bacteria. A little bit larger, the microplanktons, which include similarly photosynthetic species as diatoms, but also Réseau species as dinoflagellates. Third, we found the ciliate, which constituted essentially by intermediate Réseau species. And finally, mesozooplankton, which contain the largest Réseau species in the planktonic community, as copepods. Okay, for the methodology, this result now. First, the matter flux observed. Here you're watching the main nitrogen and phosphorus flux associated to polyester and observed in metabolic enclosure. Nitrogen at left and represented by stacking of the red and the blue bar, and phosphorus at right. As you've seen, nitrogen flux is more important than phosphorus. The first reached 1 to 48 micromolar per gram of oysters, and second is close to 0.12 micromolar per gram of oysters. Considering the nature of nitrogen flux excreted is also very important. As you've seen, ammonia represents the most important form excreted by the polyester, which effectively accounts for 19% of nitrogen flux. What is the consequences for nutrient water chemistry? Here, you've seen the nutrient balance in the farming site and the control site only for nitrogen. Phosphorus doesn't represent on this diagram. As you've seen, total nitrogen concentration represented by the stacking of the blue and the red bar is relatively similar in both sides. So, as phosphorus, which is not represented on this diagram, we don't see any accumulation of nutrients associated to performing presence. Despite any accumulation, we see an important disequilibrium in nitrogen form under performing sites. In fact, performing site favor the ammonia presence in the water column. What is the consequence for the planktons? Here you've seen the planktonic composition in the performing site than the control site with the same comparative approach. You've seen on this figure the total planktonic biomass represented by the stacking of the different bar here observe significant reduction under the performing site, about 10% of decrease. If its reduction is relatively low, it's largely associated to zooplanktonic drastic reduction observed in pale blue. Its important reduction of the zooplanktonic group is partially counterbalanced by the important promotion of phytoplanktonic group, especially the photosynthetic species, the phytoplankton, which is particularly promoted under the performing influence. As you've seen here in red. As you've seen in the results, performing represents an important source of matter flux, especially for nitrogen, for the water column. It's concomitant with previous results, especially Lacoste and Hall studies, the most recent available. And despite And despite important flux observed, we don't see any accumulation of nutrients in the water column. Its result seems to highlight the important demand of phytoplankton in the water column. In fact, our result demonstrates, as numerous studies, that lagoon is characterized by very low concentration in nutrients. Previous studies have demonstrated that the low concentration will limit severely the phytoplanktonic growth. So, we suppose that matter excreted by the polyester is so quickly assimilated by phytoplanktonic growth, by phytoplankton, and loaded in the planktonic biomass itself, limiting so the nutrient accumulation in the water column. And thus, in this scenario, matter flux seems favor fast recovering process of the planktonic community, which shown only a limited reduction, about 10%, and favor a continuous turnover. However, a closer look to our study, to our result, revealed a little bit another study. In fact, pearl farming flux favor the ammonia concentration in the water column. According to the literature, ammonia concentration is an important driver of phytoplanktonic development in the water column. It's concomitant with our results showing important promotion of phytoplanktonic group under the vicinity of farming structures. Far to be an anecdotal evidence, planktonic development is very, very important for pearl farming. In fact, phytoplanktonic is described as too small to be cleared by oysters. In this way, the matter flux generated by pearl oysters seems to promote ungrazable planktons in the water column, limiting the completion of the turnover. In conclusion, our result demonstrates an important capability of pearl oyster farming to alter the planktonic structures and likely its own food environment quality. Due to the importance of planktonic compartment in lagoon functioning, the consequences are potentially major for the world systems, but still unknown actually. In regard to our results, should we consider any possible policy management for the futures? Before to answer to this question, it appears essential to analyze the spatial and temporal variability of this alteration. How far this alteration is visible in the lagoon system? Are they present all over the years? It will be the next challenge to resolve for the future of the management of this activity. Thank you for listening.
Merci Vivien. Nous avons donc du temps pour une ou deux questions. Vous avez la parole. So we have time for one or two questions.